So good evening students. In today's lecture we are going to start with your actual syllabus of Microbial Genetics unit number 1 from the third point mutation, and from that first one is suppression mutation can in the previous lecture I have told what is the suppression mutation. Now today we will see the two basic mechanisms of this suppression mutations. There are two basic mechanism of suppression mutations, that are first one genotypic separation, second one is phenotypic separation. Now we will see first what is meant by genotypic separation. A second mutation in same genome that reverses the harmful effect of the primary mutation is called as a genotypic or genetic separation. Means what? There is one gene, in the same gene first one mutation occurs, and if the second mutation occurs, it will reverse the harmful effect which are caused due to the first mutation. It will repair and recover all the effects of primary mutation, and that is known as a genotypic or genetic separation. And based on whether the suppressor lies in the same gene or as a primary mutation or in the different gene, there are two types of genetic suppressor mutation again, based on the genes which lie on the same gene or onto the different gene. That on that basis there are again two types of genetic suppressor. And first one is intragenic separation and second one is intergenic or extragenic separation. So first we will see the intragenic separation. It is a second mutation in same gene. Only the difference is that within the same gene, the mutation occurred first in the same and second also in the same gene. The second mutation in the same gene that reverses the harmful effect of the primary mutation, that type of mutation is known as the intragenic separation mutation. And this intragenic separation mutation is again, it has three types: intracodon separation, reading frame separation, and distant compensatory separation. So we will start with first one under the intragenic separation, that is intercodon separation. Intercodon means within the same genome, within the same codon. Okay. Second mutation within the same codon, if that occurs and that will reverse the harmful effect of the primary mutation, then it is called as an intracodon separation. Means intra means within the same codon. Okay. Mutation hota, a second mutation कोणाच्यात होता तर within the same codon that will reverse the harmful effect of the first mutation. For an example, function lost by a protein after primary mutation is restored by the second mutation in the same codon. By the example we can see here, CGU is arginine. It is active protein. In this codon, first mutation occurs, primary mutation occurs, and the protein become inactive and the codon get changed into AGU serine. Okay. Into the same gene or into the same codon, again the second mutation occurs, that is suppressor mutation, and it forms AAU, that is arginine again, and it is again the active protein. Okay. Means what? And intercodon separation means within the same codon the second mutation occurs and it restores the function of lost by the first mutation. Okay. The second mutation causes restoration of original amino acid or another amino acid that can still allow the function of the same protein here. Okay. Now second one is reading frame separation. Reading frame, it first was intracodon separation. It was occurring in the same codon. Now opposite of that is reading frame separation. A second mutation within same or different codon. Within the same or into the different codon, that reverses the harmful effect of the primary mutation is known as a reading frame separation. म्हणजेच काय, एखाद्या जीनोमवरती सेम जीन किंवा डिफरंट सेम कोडॉन किंवा डिफरंट कोडॉनवरती सेकंड म्युटेशन झाला की तो काय करतो, पहिल्या म्युटेशनचा इफेक्ट रिव्हर्स करतो आणि आहे तसं पुन्हा त्याचं फंक्शन प्रोसेस करतो. Thus, frame shift mutations alters the reading frame by insertion or deletion of nucleotides. Reading frame separation मध्ये काय होतं, either insertion of single nucleotide or deletion of single nucleotide, and then it restores its function. Insertion is suppressed by deletion and vice versa. We will see with the example. As you can see here, it is a, here they have given a nucleotide sequence. Okay, this is a DNA, or this first one is a nucleotide sequence with various amino acid present in this. At the third codon site, the T is replaced with G. That is what means insertion of G codon occurs, and then it forms GTA. And by this insertion of one codon into the one amino acid, it will change whole frame of the nucleotide sequence. This is a normal sequence. Addition of one codon will change whole nucleotide sequence and it becomes out of frame. Okay, and again if we delete some of the codon from this second occurred mutation, if we delete the one codon, again it restores its own original frame. That is, restored frame again occurs, and that is known as a reading frame separation. म्हणजेच काय, एक पहिला म्युटेशनमुळं काय होतं की त्याचं फंक्शन लॉस्ट होतं, पूर्ण ती चेन काय होते, आउट ऑफ फ्रेम होते. पुन्हा सेकंड म्युटेशन झालं दुसऱ्या ठिकाणी, सेम किंवा डिफरंट कोडॉनमध्ये झालं. म्हणजेच काय, एकदा इन्सर्शन झालं किंवा डिलिशन, दुसऱ्यानंतर डिलिशन झालं. डिलिशन झाल्यानंतर सेकंड म्युटेशन झाल्यानंतर पुन्हा त्याचं काय झालं, फंक्शन रिस्टोर झालं. ओके, म्हणजेच काय, इन्सर्शन इज सप्रेस्ड बाय डिलिशन ऑर deletion and vice versa. Okay, and third one, distant compensatory separation. It is a second mutation in different codon. पहिला काय होता, second mutation in the same codon. दूसरा काय होता, second mutation into the same or different codon. It will reverse the effect of primary mutation. आता काय आहे, a second mutation in different codon that will reverse the different codon that will reverse the effect of primary mutation is known as distinct compensatory separation. It is missense mutation causing into GGG to GAG, that is glycine to glutamic acid, change in the E. coli in the formation of tryptophanase synthetase A gene resulting in the loss of enzyme activity. Due to missense mutation, means what? Missense. GGG, the G will replace with A by the primary mutation, and glycine will be converted to the glutamic acid, and by this the E. coli, in the E. coli the enzyme tryptophanase synthesize A gene result in the loss of enzyme activity. And into the second missense mutation again, UAG to UGU, that is A, again it replaced with a G, that is tyrosine to cysteine, change at a distant place 36 amino acid away in the same gene. Means from this to this there is a bit gap of 36 amino acid in the same gene, and it will again restore the function of tryptophanase synthesis activity. Means what? Neither mutation allows the synthesis of active enzyme. Okay, means what? एकदा म्युटेशन झालं, मिसन्स म्युटेशन, डिफरंट कोडॉन, एका कोडॉनमध्ये तिथं काय झालं, ई कोलायचं ट्रिप्टोफॅनेस सिंथेसिसची ॲक्टिव्हिटी काय झाली, लॉस झाली. पुन्हा 36 कोडॉन म्हणजे डिफरंट कोडॉनमध्ये 36 अमिनो ॲसिड सोडून दुसऱ्या कोडॉनमध्ये मिससेन्स म्युटेशन झालं. त्यावेळेला काय झालं, पुन्हा त्याची ट्रिप्टोफॅनेस सिंथेसिसची ॲक्टिव्हिटी रिस्टोर झाली. ओके, नाव here we have ended with the first one, that is intragenic separation, and under this we have studied 3 separations. Now B, under genotypic separation, that is extragenic or intergenic separation. It is a second mutation in different gene that reverses the harmful effect of different primary mutation. Intergenic म्हणजे काय होतं, सेकंड म्युटेशन इन द सेम जीन होता. आता इंटरजेनिक काय आहे, एक्स्ट्राजेनिक काय आहे, into the different gene. A second mutation in different gene that reverses the harmful effect which are produced by the primary mutation is known as extragenic or intergenic separation. And again this has 2 types, that is codon specific suppressor and polarity suppressor. Okay, First, we will see the codon specific suppressor. Codon specific suppressor means what? A second mutation in a tRNA gene that corrects the harmful effect of primary mutation. What? The second mutation, first mutation that occurs in the tRNA, and second mutation occurs again in the tRNA gene, different gene of tRNA, that will correct the harmful effect, and that is known as the codon specific suppressor. Codon specific means what? tRNA has a fixed codon. Into the external lecture I have told that one, and in that specific codon only occurs, hence it is known as the codon specific suppressor or informational suppressor. This codon specific suppressor are mutant tRNA molecules that have undergone a base pair substitution in anticodon. tRNAच्या इथं फक्त काय झालेलं आहे, एक बेस पेअर सबस्टिट्यूशन झालेलं आहे अँटीकोडॉनच्या ठिकाणी. tRNAला कोडॉन बाइंडिंगसाठी अँटीकोडॉन असतात आणि त्याच्यामध्ये फक्त बेस पेअर सबस्टिट्यूशन झालेलं असतं, म्हणजे एखादा बेस चेंज झालेला असतो. अँड म्युटंट tRNA अलाउज ओकेशनली इन्सर्शन ऑफ अ न्यू एमिनो ॲसिड ॲट अ साईट ऑफ म्युटंट कोडॉन. एखादा कोडॉन तिथं म्युटंट झाला की तिथं काय अलाउ करतो तो इन्सर्शन ऑफ अगेन न्यू अमिनो ॲसिड. अँड दिस कोडॉन स्पेसिफिक सप्रेसर आर अगेन ऑफ 3 टाइप्स: nonsense suppressor, missense suppressor, and frame shift suppressor. And this we will study again into the next lecture. And the second one, polarity suppressor, and the 3 types of this codon specific suppressor we will study into the next lecture. Thank you.